Hello guys and welcome back to Excel Chair Academy. So today we are going to do second lecture. So in the previous lecture of biological membrane, cell biology, we have discussed about like what plasma membrane is and what all is present on plasma membrane, right? So we have talked about like integral proteins are there, peripheral proteins are there, the carbohydrate moieties are present on your proteins, correct? And about the presence of the cholesterol. Then we have also talked about different types of phospholipids. If you remember, so we have mentioned about the PC, PI, PE, PS, right? And then the sphingomyelin. So I have mentioned before also, please do remember the composition of the the plasma membrane, and in the outer and inner layer, which phospholipid is present. So even that is crucial. So I hope you remember. And of course, the importance of the PA, that is phosphatidylserine. And we have seen its role in the case of the apoptosis. Correct. Now let's proceed. Okay. So we also discussed about like how the membrane is having the fluid-like property, correct? Where we have discussed about the saturated and unsaturated fatty acids. So we know that unsaturated fatty acids, it is going to give the kink in the structure, because of which the fluidity increases, correct? And if you have got the saturated more, so it is giving the rigid packing. So here in this case, fluidity is going to decrease. Okay. Now, so both the proteins and lipids are able to diffuse laterally through the membrane. So what does it mean? Laterally. So as I mentioned before, so we have got phospholipids like this, and if these are the numbers of the phospholipids, so after some time you will see that this one is present somewhere here, 3 is present here, right? So all this is possible. Now with this, I am able to remember about the assignment which I have given last time. So do you remember the assignment? So Chandni was not there. So Chandni can escape this time. What about others? So who can tell me what does flippase does, what does floppase does, and the scramblase? Yes, anyone?
The flippase moves the lipids from exoplasmic to the cytoplasmic area.
Okay.
So scramblases are a type of lipid energy dependent, and they can move the lipids in bidirectional way, and this is reversible also. And
Okay.
floppases move them in reverse direction, that is from cytoplasm to the exoplasmic space.
Correct, correct. Absolutely correct. So you gave all the answers for the others also. I wanted all the three to give one type. Anyway, so that is absolutely correct. So good. So I hope everyone got the difference understood over here. Chani, is it clear to you? Like what is flippase, floppase, and scramblase? Like, do you have
Yes,
any?
ma'am.
Huh? So the thing is that last time when we were discussing, so in a plasma membrane, so it is a bilayer, correct? So outer and inner leaflet is there. So we have got the outer and inner phospholipids are there. So it is possible for this phospholipid to come in this and this phospholipid to go outer. Okay. So for this, we have got the enzymes called flippase from here to here, floppase from here to here, and scramblase is the energy dependent. That's all. Simple. Okay. But you need to remember the difference so that you can answer your 2 marks question in the exam. Okay. Now coming back over here to the experiment. So this experiment was really very important, and abhi jo explain karungi, based on this only the question comes. So they are going to twist the scenario, twist the words, but ultimately they are saying the same thing. Okay. This is also one of the kind of FRAP. Okay. So this is one of the technique. Iska full form kya hoga? Fluorescence resonance after photo bleach. Yes, I think it is after photo bleach only. Okay. So isme kya hoga? That you are going to bleach a particular area of the protein. Okay. Now, when you say bleach, what will happen? What will happen to the bleach area? Are you getting? So fluorescence reactivation or recovery after photobleach. So कैसा होता है? If suppose this is your cell, आपने क्या किया है? पूरे सेल को फ्लूरेसेंटली लेबल किया है. So let us say it is green. So in the microscope, you can see that this entire cell is going to give the fluorescence green color. Agree? Now what I'm going to do is I'm going to take laser and give high intensity laser to this area. So what will happen to this area? जब आप कोई लेजर लेते हो, हाई इंटेंसिटी लेजर बीम लेते हो, एंड यू आर टारगेटिंग अ पर्टिकुलर एरिया टू अ फ्लोरेसेंट एरिया, व्हाट विल हैपन टू दैट एरिया? So it is going to get bleached. So in the case of fluorescence, if that particular area is getting bleached, what does it mean? No fluorescence. Okay, colorless. Okay, you can say colorless also. So no fluorescence or colorless. So then after some time, okay, you're incubating this cell for at least 1 hour, or like let us say 40 minutes or whatever, depending on the enzyme and depending on the cell. What do we see after 1 hour is that inside the microscope is that again the entire cell is giving me the green fluorescent. Okay, अब यहाँ पे क्या हुआ है? It is a fluorescent recovery after photobleach. Now, what exactly had happened over here? It is not that this particular area regained its fluorescence. ऐसा नहीं हुआ है. What had happened is that, let us say that this particular area, ये जो आपका bleach area है, it had around 12 phospholipids which were bleached. ठीक है? Completely bleached. Now, just imagine एक phospholipid, okay? This particular these bleached phospholipids are spread over here, like this on the entire cell, and you know that the entire cell is giving green fluorescent. अब पूरे fluorescent में, if you have got entire thing fluorescent and सिर्फ एक dot है जो आपका bleached है, do you think that is gonna make a difference? No, right? Because entire thing will be giving a fluorescence green color. So when this gets, diluted in the fluorescently labeled phospholipids, what happens? This entire thing is going to fluoresce green in color. ठीक है, समझा? थोड़ा सा आइडिया हुआ?
Yes, ma'am.
Yes, so don't worry, all these techniques will be covered when I'll be continuing with the methods in biology. So all these techniques will be covered with the animations and the figures and all. So just giving us a rough idea. Okay, so let's talk about this experiment. This is really very important to understand how they have conducted and what they concluded. So these people, Larry and the Michael, they used fusion of human and mouse cells in culture to produce human mouse cell hybrids. Okay. So then they have analyzed the membrane protein distribution using the antibody. So obviously there will be certain difference among the human cell and the mouse cell. Okay. So जो भी different protein होगा उसके against आपने antibody बनाया है. Okay. So we have used as antibody. Now these antibodies will be again labeled with the fluorescent or the chromophore. कुछ भी color हो सकता है या कोई fluorophore होगा. Okay. Now within 40 minutes after fusion, the mouse and human proteins became intermixed, and then we can see the result. So I have got a figure over here drawn only. So you can see what they have done is you have got the human cell, and the proteins which are specific to humans are labeled with red fluorescence. ठीक है? तो लेट अस से दैट ये आपका ह्यूमन सेल है, इसका जो एक प्रोटीन होगा, इंटीग्रल प्रोटीन होगा, उसको हमने एंटीबॉडी से टैग किया, एंड दिस एंटीबॉडी इज टैग्ड विद योर रेड फ्लोरेसेंस. ठीक है? सिमिलरली here also in the case of your mouse, so we have got the mouse cell, mouse specific protein, raised antibodies. Now this has got the green fluorescent. Okay, so what we have done now is that we have incubated the human cell with this antibody which is giving the red fluorescence. Similarly, mouse cells are sorry, similarly mouse cells are labeled with the green antibody. Now fusion is done. Now for this fusion, there is another video which you can follow where we have talked, where I have mentioned like there are different types of fusion mechanism. Okay, so one is electrical, that is you give electric impulse so that the membrane gets fused. Then we have got the mechanical force base. Then we have a peg base. So basically in the most of the labs, peg based fusion is done. What is peg? Anyone? Any idea what it could be?
Polyethylene glycol.
Yes, correct. It is polyethylene glycol. Good. So this is used. Okay. So mainly in the labs, we use polyethylene glycol. Now, fusion has been done. Okay. I'll share the link of this video also where I have mentioned in detail how the fusion is being carried out. Okay. So what happens? You're gonna get half of the cell giving you red fluorescence and half of the cell giving you green fluorescence. And it can be observed under the microscope also very beautifully. Okay? It's just that you kaise hoga? See, it's not that red and green both are going to give the fluorescence at the same time. This thing is also there that you need to understand. हम जब भी माइक्रोस्कोप में देखते हैं, फ्लोरेसेंट माइक्रोस्कोप में देखते हैं, okay, what happens is that we can't visualize all the fluorescence at the same time because of the difference in the wavelengths, right? And the intensity. So हम कैसे करते हैं? पहले हम red का wavelength डालेंगे and we'll take the snapshot. Okay, if suppose this is the cell and I'm giving first the red fluorescence. Okay, so मैं पहले क्या करूंगी? उसका snapshot ले लूंगी. So what will happen? This particular region is visible, but there won't be any fluorescence. ये आपको dull दिखेगा. यहाँ पे कुछ fluorescence नहीं दिखेगा. Now again, I'm gonna change the filter and I'm gonna give a command that now I need green fluorescence. हम क्या करेंगे? Green fluorescence का हम wavelength डालेंगे. Then I'm gonna get the green fluorescent का यहाँ पे fluorescence आएगा, and then this portion will look dull. Okay. Now I'm gonna take the snapshot. Now, what does the software in fluorescent microscope does is it is going to merge the image. Okay, overlap karega. Ab overlap karega. So what will happen during the overlap? I'm gonna get this region as red fluorescent and the another region as green fluorescent. So these are the images which we provide to our guide or in the research paper for the publication. ठीक है? थोड़ा सा idea मिला हम कैसे करते हैं? हाँ? So this is how. Now what happens is that ये तो हमने ले लिया एक image to show that this is our hybrid cell. So we have got red and green ka fluorescent. Okay, so I have taken the snapshots. Now I incubated the cell around 40 to 60 minutes. Then what do I see? I see that there is a mixture. Okay, so when I'm gonna give the command for red and green, I'm not going to get any good fluorescence. What I'm gonna get is the hybrid of this. So जो intermediate fluorescent रहेगा, जो overlap करने पर आता है, आपको वैसा fluorescence दिखेगा. Now, what is the conclusion from this experiment? So this is how the experiment was done to understand that phospholipids are mobile in nature. Okay. So what is the conclusion? That phospholipids एक जगह पे stay नहीं करते हैं. They are always moving. So they're always in dynamic condition. Correct? Is this much clear? This experiment is clear?
Yes.
Yeah, good. Now the question comes कि आप किसको for example exam में अगर question आता है तो वो क्या कर सकते हैं? Instead of just saying any protein, they might give you the name of the proteins to which the antibodies are attached. ठीक है? And here also they can say something, and then same experiment they will be doing and then asking like this protein was not visible or visible or what had happened. ठीक है? आप ऐसे कुछ तो वो question डाल सकते हैं, but if you understand the technique कि क्या किया है, then you can get the answer like क्यों किया है and what will be the conclusion. Okay. Okay. Now, so coming to the structure of the plasma membrane. So this is totally a different topic. So यहाँ पे अब हम क्या discuss करेंगे? Like, so these are what your integral proteins, right? So integral proteins मतलब क्या होता है? They are actually passing through the plasma membrane, correct? So these different types of integral proteins which we will be discussing now. Now एक और चीज मैं आपको nomenclature यहाँ पे समझा देती हूँ, ठीक है? So when you say this thing, so this can be your N terminal of your protein or C terminal. Similarly, over here, C terminal or N terminal of your protein. Okay. Now, when it is passing like this, can you see that zigzag? What does this indicate, this zigzag? Every time you see zigzag is there. So anyone who can tell me what does it represents? Yes, anyone? Guess? Shruti? No? Do you remember the alpha helices and the beta sheets in a protein structure? Right? So over here, this helical thing is your alpha helices. And when you say multi-pass protein means एक आपका protein है, let us say this is N terminal, this is C terminal. So ये एक protein है जो आपका alpha helices बन रहा है, making a turn, then making over here again like this and like this. So this means what? The transmembrane proteins, they are also known as transmembrane proteins. और ये कितने बार span हुआ है? 3 times. So if suppose someone is asking that you have got, let us say GPCR is there. So what is GPCR? Your G protein coupled receptor, correct? Which is used for the vision, taste, etc. Now, GPCR is having 7 membrane spanning domain. So what does it mean? It means that GPCR में ऐसे आपके 7 रहेंगे. ये 3 हो गए, 4, 5, 6, 7 ऐसे रहेंगे. ठीक है? This is the multi-pass protein. Now, when you say multi-subunit protein, this is single हो गया. ये आपका multi हो गया. It is a single protein only. Here also single, but it is passing the plasma membrane 3 times. Okay. It is also known as spanning the membrane 3 times. Now, what about this? Here, subunit protein, multi-subunit protein. So, N terminal here, C terminal C. यानी कि यहाँ पे क्या हो गया? More than one protein you are having. ठीक है? More than one protein which is spanning the plasma membrane. Then you have got the peripheral जो आपके नीचे ऐसे hang करते रहते हैं. Is this much clear? Guys, Chandni, Tanvi? Okay.
Ma'am?
Yeah.
Shruti wanted to ask something, but she's not able to speak. Like she's not audible.
Okay,
so she wanted to leave and join.
No, maybe she can message if I'm audible, then that's fine. Otherwise, she can chat, put into the chat.
Okay.
Huh? If she's not able to hear, then tell her to rejoin then. Okay, Shruti. So, I mean, if possible, you can type your question over here. I'll read out and then explain. We'll just wait for her. What about others? It's clear?
Yes, ma'am.
Okay. Okay, so her question is why to use laser? White light will allow both colors to be seen simultaneously, right? White light will not give the fluorescence, dear. See, in the fluorescence microscopy, when we apply, okay, so if I give white light, what will happen? Everything is visible then, correct? It is not going to give me the difference between the red fluorescence and the green fluorescence. I need to provide the machine to give the wavelength. Acha, I'll give you another example. Wait, I'll explain. If you know something about the Stokes shift, if you have heard about it. Okay, so what does it? It says, I'm gonna give certain wavelength light so that the fluorophore is going to absorb, right? When it gets absorbed, it gets energy, it gets excited. And then when it is coming back, it is going to give emission. So the absorbing wavelength is different and the emission wavelength is different. So when I say that I need red fluorescent, it doesn't mean that the red wavelength I'm giving. I'm giving a wavelength of lower range. Okay. So when I give less, red means around 600 nanometer, right? Here I will be giving around 455 nanometer. So the fluorophore is going to absorb this and going to emit this. So if I give white light, what will happen? It has got the combination of all the wavelengths. So it is not going to absorb. The machine is not able to provide a single light. It has to be a monochromatic light. I hope everyone knows what is monochromatic, right? So when I give, then only I'm gonna get red. अब व्हाइट लाइट में आपको सब दिखेगा, मगर वहां पे क्या होगा, आपका जो फ्लूरोफोर है, it cannot absorb, it cannot get excited, and that's why we need to give monochromatic wavelength laser, right? Okay, is it clear, Shruti? Okay, because here we are talking about the fluorescence. अगर मैं यहाँ पे आपको बोलती कि आपने कुछ क्रिस्टल वायलेट डाई यूज किया है व्हाटएवर, देन वी गो फॉर व्हाइट लाइट, लाइक नॉर्मल अपना ब्राइट फील्ड माइक्रोस्कोप होता है, राइट? बट इट इज नॉट गोइंग टू डिस्टिंग्विश. क्रिस्टल वायलेट वाला कॉन्सेप्ट हम यहाँ यूज नहीं कर सकते बिकॉज आई एम टारगेटिंग अ वन स्पेसिफिक प्रोटीन. व्हाट डज क्रिस्टल वायलेट डज? इट इज गोइंग टू स्टेन द एवरीथिंग. And whereas in the case of the mammalian cell, it is going to kill the mammalian cell. वो उसके लिए टॉक्सिक हो जाता है. So that concept we cannot use. Okay. Yes. Okay, so now let's talk about the membrane associated proteins. Already mentioned that either they are going to be integral or peripheral. Okay. Now integral proteins, so they are the amphipathic proteins. So what does amphipathic means? Both polar and non-polar regions are present. So non-polar आपके ये region हो गए, ये आपके polar region हो गए. ठीक है. Now this needs to be, if suppose you need to purify this integral proteins or membrane proteins, okay, if you need to purify for your experiment, then how you're going to do? So if you remember, we have talked about the cell lysis during the techniques, methods and all, right? So if suppose I am taking animal cell and I need only the membrane bound. So मैं क्या करूंगी? Animal cell rupture करूंगी. I'm gonna remove the cytoplasm. I don't want cytoplasm. क्यों? Because cytoplasmic proteins are gonna interfere with my purification of membrane proteins. I want only membrane proteins. So I'm gonna take the cell membrane only. Now it is going to have the plasma membrane components like all the phospholipids, cholesterol, as well as the proteins. Correct? I am interested in proteins. So now I'm gonna use detergents. I have also mentioned that there are certain ionic, non-ionic detergents are there. So there is something, for example, Triton X10 is there, X100 is there. So all these mild detergents are there, which I can use to purify the plasma proteins or the membrane bound proteins. Okay, why? Because you know that the detergent also have got both polar and non-polar region. So if my protein is also having polar and non-polar region, the detergent is going to associate the protein in such a way that polar में polar रहेगा, non-polar में non-polar रहेगा. So then it becomes easy to remove the proteins from the phospholipids. ठीक है? Eventually, I will just use only the detergents so that I can purify the protein. ये एक step हुआ, उसके बाद you can go to the chromatographic techniques which we'll discuss later. इतना clear है? If anyone is asking like how do you purify your membrane associated protein, simple answer is by using the detergents, okay, mild detergents. Okay, so some integral proteins are the transmembrane proteins extending completely across the membrane, यानी कि पूरा ऐसे जाएंगे. Hydrophobic alpha helices are common, so these are your hydrophobic alpha helices. Some wind back and forth across the membrane. So this is the example जहाँ पे आपका C terminal है, then वापस आया है, then ऐसे गया है, then वापस आया है. Few are like simply just like this. तो एक सिंगल रहेंगे. ओके, सो बेसिकली इफ इट इज अ सिंगल देन इट इज वर्किंग एज ए रिकग्निशन रिसेप्टर, जहाँ पे आपको सिग्नल देना है या रिसीव करना है. और अगर मेरे पास ऐसे मल्टी स्पैनिंग अल्फा हेलाइसिस वाले अगर ट्रांसमेम्ब्रेन प्रोटीन्स है, तो बेसिकली दे एक्ट एज ए रिसेप्टर, लाइक जहाँ पे लिगेंड आके बाइंड करेगा एंड देन इट विल गिव सर्टेन अदर सिग्नलिंग. ओके, now pore protein, this is, so this, look at the structure and tell me what is the main difference over here. This is also one of the plasma membrane, but there is a huge difference in the structure. Now who can tell me the huge difference? सबसे बड़ा difference है, this is the exceptional case in the case of the transmembrane proteins, pore protein. So हम इसके बारे में आगे पढ़ेंगे. Pore proteins like you have got those porins are there, जहाँ पे बैक्टीरिया pore protein release करता है and it is making your cell to lose the entire cytoplasmic content. So जहाँ पे dehydration, loss of water होता है, and that's how bacterial toxins are actually affecting. जहाँ पे हमें diarrhea हो जाता है या कुछ हो जाता है, right? So these are those pore proteins. Now look at the structure, look at the figure, and tell me what is the huge difference between this transmembrane protein and the one which we have just seen now.
This has beta sheets, and that one, the transmembrane protein, that was made up of alpha helices.
Correct, absolutely correct. So this is the exception where you can see that the pores or this protein structure is actually is only made up of beta sheets. Whatever the rest we are going to discuss now is going to have the alpha helices. Now what happens is over here is कोई भी बैक्टीरियल टॉक्सिन आया, ठीक है, लाइक आपने कुछ गलत खाना या स्पॉइल फूड खाया या व्हाटएवर द बैक्टीरिया इन्फेक्शन यू गॉट, it is going to release this pore protein. Okay, it's a toxin actually. And हमारे intestine में जो भी आपके जो cells हैं, okay, absorbing cells, you have got ऐसे cells रहेंगे which is supposed to absorb all the nutritions from the food what you have taken, right? So these pore protein, they're gonna make hole in this. It's like a pore. ऐसे पोर बना देगा या ये नॉर्मल हो जाएगा पोर. ओके, it makes it hollow kind of pore. What will happen then? Entire cytoplasmic content of this will go outside. ठीक है, एंड ये हमारा हो जाता है लूज मोशन या लॉस ऑफ वाटर, रेगुलर डिहाइड्रेशन, वॉमिटिंग, ये इन सब में हमारा पूरा न्यूट्रिशन चला जाता है. So this is how it is affecting us. Now suppose any doctor is giving the medicine like to prevent the diarrhea or the loose motion, so it is going to target this pore, it is going to block it. Okay, and that's how we recover from that. If you remember, हम हमेशा बोलते हैं ना अगर डायरिया हो या लूज मोशन हुआ, वॉमिटिंग ज्यादा हो रहा है, so we always ask them to take ORS, correct, ग्लूकोज लेने के लिए बोलते हैं, because everything you have lost. Okay, so इसलिए हम बोलते हैं इसको पीते रहो till you become normal. तो डिहाइड्रेशन नहीं होना चाहिए, otherwise it is going to affect the brain activity. Okay, clear? Okay, now about the integral monotopic proteins. Okay, so they are the proteins which attach to only one side of the biomembrane. Can you see the difference? ये हमारा inner side हो गया है. Okay, यानी कि इस तरफ आपका साइटोप्लाज्म है, उस तरफ आपका एक्स्ट्रासेलुलर स्पेस है. Okay, examples are glycophorin, which is mainly present on RBCs. They are rich in sialic acid, giving negative charge to RBC. So here, see, take away message कितना सारा हमें मिला यहाँ पे. So what is it? Glycophorin, they are present on the RBCs, rich in sialic acid, and they are acid, right? So negative charge. So it is going to give the negative charge to your RBCs. Now, why does the RBC have got the negative charge? नेगेटिव चार्ज की क्या जरूरत है उसको? Yes, anyone? Yes, anyone? Tanvi, would you like to try? Question is why? Okay, we know that RBCs are now negatively charged. This negative charge is given by sialic acid. My question is why there is a need for RBCs to be negatively charged? कोई तो reason होगा. That's why the nature has made our RBCs like that, right? So I hope question is clear. Yes. Anyone? Chandni, would you like to try? Akshaya?
Is it for the hemoglobin to attach?
ना. हीमोग्लोबिन तो आपका आरबीसी के अंदर हो गया. This is the RBC का surface. Outer surface negative charge दे रहा है. Why? No one? Shruti, would you like to try? Acha, your mic is not working. Anyways, so it's very simple. So we all know that in blood vessels, okay, RBCs are continuously moving in big number. ऐसा तो है नहीं कि सिर्फ एक टाइम पे एक आरबीसी जाता है. It is like so many RBCs are going together. Obviously, the chances are that the RBCs, one cell is going to match, attach to the another one. If suppose this negative charge was not given, okay, if this negative charge was not there, then because of this protein-protein interactions, the RBCs would interact with all each other and forms the aggregate. Then the then the RBCs are not able to conduct its activity, right? वो एक जगह से दूसरी जगह नहीं जाएगा, वो ब्लड वेसल्स में एक ही जगह इकट्ठा हो जाएगा, एग्रीगेट बन के फॉर्म होके नीचे बैठ जाएगा. Are you getting the point? Now, if sialic acid is there and it is giving negative charge to RBC, so what is happening? It is creating a repulsive electric zeta potential on the surface of the RBCs. What did I just mention here? The electric zeta potential we say. Normal भाषा में क्या? It is giving a repulsive potential to each surface of the RBC. Now, because of which, जब भी कोई भी RBC दूसरे RBC के पास आता है, because of the negative charge, what will happen? So both the cells are having negative charge, so they are going to repel. Correct? And because of this repulsive forces, they are not getting attached or forming the aggregates. So this is the reason why we need negative charge on the RBCs. Clear? Now it is clear?
Yes, ma'am.
Okay, so these are important, small points, but it is important. Okay. Okay, so now, okay, so this makes them to circulate without disturbing the other cells. Okay, so then we know that the glycophorin, there are different types are there, and this is a glycophorin which is responsible for the malarial attack on us. So we'll discuss about that also later. Now look at the structure here. So what do we see? You have got the plasma membrane, right? And glycophorin is present over here. Okay, now glycophorin is there. Now can you see the attachment of the glycophorin to the rest of the proteins? Now every time, as I mentioned, सबसे पहले जो discovery हमने की थी प्लाज्मा मेम्ब्रेन की RBCs पे की थी, right? So after that, main study or the analysis of various integral and peripheral proteins was also done using the RBC's membrane only. Okay. Now, if you see in the structure, what all do we see? So we saw that glycophorin is there, band 3 is also there. Okay. Please do remember about the band 3. One second. Yeah. So from here onwards, you need to remember all this thing, all the proteins name which is written over here. So glycophorin हो गया, इसको एक छोटा सा तरीका याद रखो that it is associated with our malarial attack. Band 3 is responsible for the exchange between oxygen and CO2 in the lungs. ठीक है? Then all these ankyrin हो गया, tropomyosin हो गया, edusin हो गया, tropomodulin हो गया. So they all are your supporting proteins. Then spectrin also, there's something which you need to remember. अब हम धीरे-धीरे जाते हैं what all these proteins are doing. ठीक है? So you have seen glycophorin का काम, band 3 also I have mentioned. Now let's look at the structure. So this is your glycophorin ka part, lower part. It is associated with your actin proteins. Now, what are actin proteins? If you remember, those are nothing but which forms your G actin, which later becomes your F actin. तो ये जो बना है आपका, वो आपका F actin है, filamentous actin from globular actin. Now, what is actin? It is also a part of cytoskeleton structure, right? We have got 3 cytoskeleton structure: mitotubules, intermediate filaments, and the actin filaments, micro filaments. Okay, micro filaments like that. ठीक है? इतना clear है actin? What is actin? Okay. Now, next is tropomyosin. Tropomyosin. So this tropomyosin is bringing your actin together, and then you have got your band 4.1, edusin, tropomodulin. What all they are doing? If you see carefully, band 4.1 जो है आपका spectrin actin को join कर रहा है. Spectrin ये green color का जो है, ठीक है? तो band 4.1 का काम है to bring your actin and spectrin together. Then edusin का क्या काम है? To bring spectrin, actin, tropomodulin. Okay, ये आपको support दे रहा है. Now, keeping all this thing in the mind, I'm going to now explain how to understand what is happening over here. Okay, so imagine this is your cell. This is 3D, right? यहाँ पे बहुत space है. Now, you need to keep this membrane holding over here. जैसे हम क्या कहते हैं, कोई घर की दीवार को होल्ड करने के लिए चाहिए, राइट? वो होल्ड कहाँ से होगा? व्हेन यू गिव देम दी बेस, पिलर आप जो देते हो. सो दैट पिलर इज फॉर्म्ड बाय दीज एक्टिन फिलामेंट्स. एक्टिन फिलामेंट्स ऐसे से बने रहेंगे व्हिच इज होल्डिंग योर प्लाज्मा मेम्ब्रेन. अब उसको एक्टिन फिलामेंट्स एंड प्लाज्मा मेम्ब्रेन को कनेक्ट करने के लिए यू नीड लिंकर्स. जैसे सपोज ये आपकी दीवार है एंड वी कीप इट ओवर हियर बेसिस, राइट? जैसे हम कभी कोई दीवार बनाते हैं ऊपर की, लाइक द टेरेस व्हेन यू मेक, यू कीप योर बैम्बूज ओवर हियर, राइट? बैम्बू रहेंगे, फिर वो वहां से आपको ये सपोर्ट देते हैं. इसके ऊपर ही आप सीमेंट डाल के आप छत बनाते हो, करेक्ट? तो सेम चीज ओवर हियर एक्टिन एंड दीज प्रोटीन्स आर डूइंग. नाउ व्हाट डज स्पेक्ट्रिन इज डूइंग? अगर आपका एक्टिन फिलामेंट्स ऐसे छत पकड़ रहा है तो स्पेक्ट्रिन इन दोनों को कनेक्ट कर रहा है, ठीक है? अगर यहाँ पे कुछ पकड़ रहा है तो वो उसको ऐसे कनेक्ट कर रहा है. So spectrin is making sure that the actin filaments are interconnected, okay, and actin filaments are holding the cell membrane so that it doesn't collapse. Otherwise, what will happen? 3D से आपका ये 1D हो जाएगा, पूरा चपाट हो जाएगा, एकदम फ्लैट हो जाएगा, ठीक है? Now, what does this tropomyosin, band 4.1, edusin, tropomodulin are doing? They are holding the spectrin and the actin का connection, okay? So this is how it is helping the entire cell to hold its structure, to remain in the 3D form rather than becoming 2D or 1D. Is it clear? Yes? Imagine हो रहा है या should I explain once again? Because yeah, there is one video is there, but it won't clear. It is not that clear, like how I really want to convey the message. But are you able to follow like how these proteins are helping in holding the 3-dimensional structure of the cell? Yes, Shruti?
Yes, ma'am.
Okay. Akshay, Chandni, Tanvi, are you able to follow? ये धीरे धीरे और clear होगा because eventually at the end of the cell biology lectures you will be remembering all these proteins.
Yes, ma'am.
Okay. So it's okay. I mean, it's just one time only, like first time you're hearing. So obviously won't be able to remember. I just want you to understand the concept. Okay. Now let's come to the band 3. So what is band 3? Here I said it's already a dimer. So when I say it's a transmembrane protein, so what does it mean? It is going to cross a plasma membrane. So when it is at 14, what does it mean? It का इसका मतलब है अगर ये आपका एंड टर्मिनल है तो ये आपको 14 बार पास करना होगा ऐसे ऐसे से 14 times it is going to pass the plasma membrane, okay, encoded by this carrier gene, solute carrier family 4-member gene. It is also known as anion exchanger 1. It is also known as the biocarbonate channel. Biocarbonate anion exchanger. Biocarbonate क्या है आपका? Correct? It is negative charge. So it is also known as anion exchanger. Many activities like protein anchor, transporter activity, chloride transport, etc. are occurred by this protein. So even though it is having this name, it is responsible for many functions. And we'll talk about this band 3 in detail. Don't worry about it. ये तो बहुत ही important है. Now let's talk about the peripheral protein. So अब तक क्या था? A glimpse of like what kind of integral proteins we will be discussing. Now coming to the peripheral proteins, usually they are bound ionically or by the hydrogen bonds to a hydrophilic portion of the integral proteins. Now, peripheral proteins के example. Same diagram if you see, but different proteins we are going to discuss now. So here, ankyrin and band 4.1 is your peripheral proteins. Okay, ankyrin क्या काम करता है? It's a mediator protein between the spectrin and the band 3. ये जो green color portion है ये आपका spectrin है. There are two types of spectrin, spectrin A and B. उसका tetramer बनता है. Okay, ankyrin. So it is going to link the plasma membrane and the cytoskeleton. Okay. Now band 4.1, it maintains the structure and skeleton of the RBCs again because it is helping in maintaining the 3D structure of the protein, structure of the cell membrane. It is responsible for that. Okay, yeah. Now, so the protein profile of one membrane side typically differs from the other side of the cell. So here also it is important to understand जब हम कोई बोलते हैं ऐसा cell है तो ये portion of plasma membrane is going to be different from this portion, is going to be different from this portion. So what does it mean? It does not have the same layer. ठीक है? Also, when they were doing the freeze fracture technique, okay, which we'll be discussing soon, in that what they have seen is that the extracellular layer and the cytoplasmic layer, the difference is seen. Now looking at this image, okay, this is electron micrograph image. What do you understand? Suppose आपने तो टेक्निक कर लिया, ओके, अब हमने देखा ये आपका हो गया एक्स्ट्रासेलुलर लेयर ऑफ द प्लाज्मा मेम्ब्रेन, ये होगा आपका साइटोप्लाज्मिक लेयर. अगेन थोड़ा सा और क्लियर कर देती हूँ, ये आपका प्लाज्मा मेम्ब्रेन है, ये वाला आपका ये दिखा रहा है, एंड दिस वन इज योर दिस वन, ओके, in the electron micrograph. Now looking at the figure, okay, looking at the image, what conclusion can be drawn? Yes, Tanvi, will you try to give answer? बस यहाँ पे देखो क्या अंतर आ रहा है. This image and this image. Anyone, it's not like only on the pressure is on Tanvi. Anyone can answer. See, this is how you're going to develop your analyzing or analytical thinking, right? The figure is given, you know the technique. हमने क्या किया है, okay? तो दोनों figure देख के आपको क्या समझ रहा है? Like what exactly is happening? And we are talking about proteins. Akshay, would you like to try?
is the cytoplasmic membrane layer showing the proteins which are embedded inside?
Not exactly embedded it, but what does it signifies? Okay, you're going on the right track, but figure देख के क्या लग रहा है, क्या हो रहा है दोनों केस में, to the outer layer and the inner layer, क्या हो रहा है? You are on the right track. It is all about the proteins, but conclusion क्या है? Yes, Chandni, would you like to try? Shruti?
There are more protruding proteins on the cytoplasmic layer.
Yes, correct. It's not the protruding, but it says that cytoplasmic layer has got more protein as compared to the extracellular layer. ठीक है? Why? I mean, protruding is not the correct word, but integral. Correct? See, अभी क्या होगा? If suppose ये outer है, तो यहाँ पे हमें सिर्फ integral proteins जो बाहर निकले हुए उतना दिखाई देगा. But inner में क्या है? You have got integral also as well as peripheral also. So from this you understand that the more proteins are present on the cytoplasmic layer as compared to the extracellular layer. बस इतना ही था, simple, right? But this all should be clear when you look at the image. तो आपने ये देखा, you can see that extracellular, so many small, only few dots are there. So those are of the integral proteins. Here cytoplasmic, it is both integral proteins as well as the peripheral proteins, and that's why you see so many dots are there. So अब इससे भी आपको क्या समझ रहा है? That the cell membrane of the cell is having so many millions of proteins are present, right? And this regulation of each protein is so crucial for the survival of the cell. It's not like only one or two are there, many proteins are there, correct? And it is the duty of the cell to understand, to get the proper signal from the outside and to convey to the inside of the cell. Okay, so only this much is the conclusion, that cytoplasmic layer has got more proteins because of the presence of the integral as well as the peripheral proteins. Okay, clear? Any doubts so far? Any other doubts are there? Akshaya, Chandni, Shruti, Tanvi?
No, ma'am.
Okay, cool. So I'll be stopping here now and we'll continue in the evening with the same.